Introduction to Histology, Tissue Processing and Microscope. Dr. Abubakar H. Mossa. Anatomy Department. 8 and the 11th of September 2011. Lab 1. Outlines, Definition of Histology, Basic Organization of Human Body. Tissue Processing. Fixation. Dehydration. Clearing. Embedding. Sectioning. Staining. Light microscope, parts and how to use. Definition of histology. Histology is the study of the tissues of the body and how these tissues are arranged to constitute organs. The Greek root histo can be translated as either tissue or web. Histology involves all aspects of tissue biology, to correlate the tissue structure with its function. Basic organization of human body. Tissue processing. To be able to study the microscopic features of the tissue, it should undergo several steps. Each step is done for a certain reason. Here, we are going to talk about these steps briefly. Tissue processing. Tissue processing fixation. The most commonly used substance is formalin. Aqueous solution, hydrophilic. Used to preserve the tissue by terminating cell metabolism, preventing autolysis, sterilizing the tissue, hardening the tissue, tissue processing, embedding. After hardening, fixing the tissue, we have to put the small pieces of the tissue in a block of paraffin, embedding, to facilitate the next step, sectioning. But we cannot embed the tissue in paraffin without washing the tissue from formalin. Then drying it, dehydration, by using alcohol. Finally removing the alcohol, clearing, by organic solvents. The tissue, then, is immersed in hot paraffin and left to cool to obtain the tissue blocks. Tissue processing sectioning. This is done by a machine called microtome. The blocks are sliced in the range of 5 to 15 micrometers, 1 micrometer equals 1/1000th of a millimeter. The slices then are fixed on their slides. Sectioning. Tissue processing staining. If the slide is examined under the microscope without staining, we can see only vague shadows of the tissue. So to visualize the tissue and the cellular components, we have to use dyes which stain the different components according to their chemical composition. Tissue processing staining. Different dyes or stains are used to display the cellular components. Hematoxylin and eosin, H&E, are the stains used in most of the slides we are going to study in our course. Eosin is an acidic dye and stains the basic cellular organelles in blue. Hematoxylin acts like basic dye and stains the acidic components in different degrees of red color. H&E stain. Other stains. Other stains. Other stains. Tissue processing. Rough endoplasmic reticulum. Parts of microscope.